If you could look inside a cell, you'd see a landscape in constant motion. Vesicles glide along invisible highways, organelles shift position, and chromosomes prepare to divide with astonishing precision. The secret behind this organization is a microscopic structure called the microtubule, a hollow, flexible tube that serves as both the scaffolding and the transport network of the cell. But unlike steel beams in a building, microtubules are alive with activity. They're constantly rebuilding, breaking, and building themselves in response to the cell's needs. They don't just hold the cell together, they guide its every movement, division, and internal traffic flow. In this video, we'll explore how these incredible molecular tubes are formed, how they move materials across a cell, and how understanding them has revolutionized medicine, particularly in cancer treatment. Microtubules are hollow cylinders made up of repeating protein subunits called tubulin dimers. Each dimer contains two smaller proteins, alpha-tubulin A and beta-tubulin B, that join together to form the structural building blocks of the microtubule wall. These filaments radiate outward from a central structure near the nucleus called the centrosome, which acts as the microtubule organizing center. From there, they spread throughout the cell like spokes from a wheel, giving the cell its internal organization and shape. Microtubules are not rigid, they constantly grow and shrink, a property known as dynamic instability. This constant remodeling allows cells to reorganize instantly in response to internal and external signals. For instance, forming spindles during cell division or directing vesicle transport toward the cell membrane. So while they may seem like simple structural elements, microtubules are actually living scaffolds that define how cells look, move, and function. Let's zoom in to the molecular level. Each microtubule begins with alpha and beta tubulin dimers assembling end-to-end into long chains called protofilaments. 13 of these protofilaments align side by side to form a hollow cylindrical tube, about 20 nanometers in diameter, large enough to be one of the most visible structures under an electron microscope. Every tubulin dimer binds to a molecule of GTP, guanosine triphosphate. GTP acts like a biochemical battery, providing energy that drives the assembly and disassembly of the microtubule. Microtubules have polarity, a plus end and a minus end. The plus end grows faster, adding GTP-bound tubulin dimers, while the minus end, usually anchored at the centrosome, is more stable. As GTPs hydrolyze to GDP, after incorporation, the structure becomes less stable, which is what allows it to eventually depolymerize. This push and pull balance of growth and breakdown gives the microtubules both strength and adaptability, a molecular dance of construction and collapse. One of the most fascinating features of microtubules is dynamic instability, a behavior where microtubules alternate between phases of growth, pause, and rapid shrinkage. When the microtubule has a cap of GTP-bound tubulin at its growing end, it's stable and continues to elongate. But when that GTP is hydrolyzed into GDP, the structure destabilizes. The end peels back like unraveling tape, leading to a rapid collapse known as a catastrophe. Sometimes a microtubule can rescue itself by adding new GTP tubulin dimers before total disassembly, restoring its stability and beginning growth again. This constant back and forth growth catastrophe rescue is how the cell maintains a dynamic yet organized cytoskeleton. Dynamic instability is more than just movement. It's the essence of cellular flexibility. When a cell migrates, divides, or reorganizes, this rapid remodeling allows it to respond within seconds. Without it, cells would be rigid, static, and unable to adapt to the ever-changing demands of life. Microtubules aren't just for structure. They're also the highways of the cell. Along these protein filaments, tiny molecular machines called motor proteins carry cellular cargo back and forth with incredible precision. Two major proteins handle this logistic system, kinesin and dynein. Kinesin moves toward the plus end, carrying vesicles, proteins, and organelles toward the cell membrane. Dynein moves toward the minus end, transporting materials back toward the nucleus. Each of these proteins literally walks along the microtubule using energy from ATP hydrolysis, taking 8 nanometer steps, one tubulin dimer at a time. Under a microscope, it looks like two tiny legs walking hand over hand, towing a vesicle behind like a train car on a rail. This system ensures that organelles like mitochondria, endosomes, and lysosomes reach their correct destinations. It's so precise that disrupting microtubule transport can impair neuron communication, muscle contraction, or even hormone secretion. So the next time you imagine traffic jams, think of the cell's highways, flawlessly organized and perpetually in motion. When a cell prepares to divide, its microtubules take on a new critical role, organizing the mitotic spindle, the machine that ensures each daughter cell gets the right set of chromosomes. At the beginning of mitosis, microtubules disassemble and then reassemble into spindle fibers extending from centrosomes at the opposite poles of the cell. These fibers attach to the chromosomes at specialized protein complexes called kinetochores, located at the centromere. Once attached, the microtubules exert force pulling sister chromatids apart and guiding them toward opposite ends of the cell. This process requires exquisite timing and precision. Any failure here can lead to aneuploidy, an incorrect number of chromosomes, which is one of the hallmarks of cancer. After the chromosomes are separated, microtubules disassemble again, ready to rebuild the interface network once division is complete. In short, without microtubules, cells couldn't divide, develop, or reproduce life accurately. They are the architects of order during one of the most chaotic events in biology. Because microtubules are so essential for cell division, they become powerful targets for many important medical treatments, especially in cancer therapy. Drugs like Taxol work by stabilizing microtubules. This means they prevent microtubules from breaking down during division. When that happens, cancer cells can't separate their chromosomes properly, so they stop dividing and eventually die. Other drugs like colchicine do the opposite. They prevent microtubules from forming in the first place, which also stops cell division. These two approaches, freezing or preventing the formation of microtubules, are both effective ways of slowing uncontrolled growth of cancer cells. But microtubules are important far beyond cancer. In the brain, they help maintain the structure and function of nerve cells. When the microtubules break down, such as when the tau protein becomes defective in Alzheimer's disease, neurons lose their stability and eventually die, leading to memory loss and cognitive decline. So studying microtubules doesn't just teach us how cells divide, it helps us understand and fight some of the most serious diseases known to medicine.